Good morning, I'm Fauzi Maluf Filho, Associate Editor for Gastrointestinal Endoscopy, and today I have the privilege to interview Michael Wallace, Editor-in-Chief of Gastrointestinal Endoscopy from the Department of Gastroenterology and Hepatology, Mayo Clinic, Florida, Jacksonville, who has recently published his research entitled A Novel Submucosal Injection Solution for Endoscopic Resection of Large Colorectal Lesions, a randomized double-blind trial. Mike, thanks for coming. Mike, this research was a 5-center randomized control trial comparing the injection of a submucosal solution named SIC 8000, commercially known as Eleview, with the injection of saline for endoscopic mucosal resection of sessile colorectal polyps larger than 2 cm. The main outcomes were the total injected volume and time of resection. 226 patients were enrolled and 211 entered in the final analysis. Could you summarize the main findings of this trial?
Good. Thank you, Fauzi. Before we begin, I just want to make a brief comment on, this was an unusual role for me because I was an author on this paper, but also it came to our journal where you and I are both editors and associate editors. In that situation, we actually send these manuscripts to an independent editorial board. We're fortunate to have an independent board, so they judge these manuscripts so we can avoid any, or at least minimize any conflicts of interest. But I'm very happy that the paper was published in Gastrointestinal Endoscopy. I'm very proud of our journal and really want to publish the very best papers in the field of endoscopy and in gastrointestinal endoscopy. In terms of summarizing this study, so this was a large study. It was a 5-center randomized double-blind placebo-controlled trial. Actually not placebo. The control arm was standard saline methylene blue injection. The intervention arm was a new viscous agent that contains methylene blue, saline, and also an agent that becomes viscous when you inject it. We know previously that viscous agents, when we inject these for EMR, improve the efficiency of the procedure. It allows us to resect more quickly, in fewer pieces, more en-bloc resections, etc. So this was a new available agent. The main advantages potentially of this agent were that it would improve the efficiency of the procedure. Our main hypothesis was really focused around the efficiency parameters, the time of resection, the number of pieces, the size of the resection pieces, not so much the clinical outcomes. We didn't think that there would be a difference in the complete resection rate. So we focused on clinical outcomes. Our main primary aims were the time for injection and the time for the procedure. That was significantly shorter with the newly available agent. It goes by a long chemical name. Its trade name currently is Eleview. We also found an improvement in the so-called Sidney resection quotient. This is a parameter that measures the size of the pieces that we resect. Secondarily, we looked at adverse event rates. We didn't see any difference in adverse event rates overall. So overall, it was a positive study in that the new agent was more efficient, similar efficacy, and no difference in adverse event rates.
Nice. And could you characterize the polyps that were resected in terms of mean size, location, and Paris classification?
So these were all large colorectal lateral spreading lesions. The entry criteria was that it had to be at least 2 centimeters in size, and that was objectively measured using a 2 centimeter snare. We had to photograph the lesion adjacent to an open snare to document its size precisely. So all of the lesions were at least 2 centimeters to get into the study. The mean size of the lesion that was actually enrolled was 33 millimeters or 3.2 centimeters. Most of these were proximal colon lesions. About 3/4, 74% were located in the proximal colon. And as with most of these lateral spreading lesions, the majority were Paris Class 2A. 58% were Paris Class 2A. 34% of them had a 1S or nodular component. So these are your typical granular lateral spreading polyps that I think all of us are seeing increasingly in our practice.
Nice. And did you include lesions with the previous attempts of EMR?
No, we actually excluded, if someone had done a previous hot snare resection or attempted an EMR, these were excluded because we know that that significantly impacts the ability, the speed, the proficiency, and even the safety. So we wanted to exclude that group.
Okay. And would you say that the findings are consistent regardless of polyp location or Paris classification?
Generally speaking, yes. We did not power the study, and it was not a pre-specified aim of the study to look at efficiency in various locations or in various sizes. But broadly looking at it, the results in terms of the improved efficiency and safety were broadly applicable to polyps of all size, generally polyps of all shape and location.
And which adverse events should we expect with this new solution?
So the bottom line was the adverse event rate was similar in both the standard saline methylene blue arm as well as the Eleview, the viscous agent arm. So we saw about 10% or 8 to 10% adverse event rate. It was actually slightly lower in the viscous agent arm, but there was no statistically significant difference between the two. The most common adverse event was intraprocedural bleeding, followed by delayed bleeding within the first 24 hours. We measure that up to 30 days, and we did a phone call to every single patient at 30 days to capture additional adverse events. We had one perforation in the trial. It was in the saline arm that was clip closed and managed medically.
And do we need a special needle to inject this viscous solution?
We don't. Actually, one of the interesting physical properties of this agent is it's not viscous as it's being injected. It becomes viscous within the tissue. So it cross-links collagen and other components in the submucosa. So when it's injected, it's injected with the same viscosity as saline, but once it comes in contact with tissue, it cross-links within that tissue and becomes a very viscous agent.
So basically you use a standard 23, 25
Correct. Any needle that you're currently using is appropriate.
Okay. And final comment, could you comment on the impact of your research for those who perform colorectal EMR, including possible cost impact? Or
So as you know, it's currently the way we prepare our EMR solutions is somewhat dependent on the center. This is the first FDA-approved pre-mixed viscous agent that is indicated for endoscopic mucosal resection. Everything we've been using to date was really mixed on site. We typically took combinations of methylene blue, saline, some viscous agent. Almost all endoscopists have their own mix. We often have to ask our nurses or pharmacists to mix that on site. And so there hasn't been a good standardization. So some of the advantages of this are the standardization. It's obviously FDA approved for this indication. It was developed specifically for this indication. It comes pre-mixed, so there's no requirement for, say, a compounding pharmacy or asking our nursing assistants to prepare it. In terms of cost, the major drivers of cost, I don't know exactly what the cost of the agent is. We have looked a little bit at the cost of our current agents. The main cost of our current agents is the methylene blue, which is rather expensive. In our pharmacy at Mayo Clinic, it's about $150 per vial, and that's a single-use vial. So the Eleview agent used in this trial, I believe is quite comparable in terms of cost, at least until you get up to 1 or 2 vials of it. At those levels, it would actually be less expensive than our current premixed methylene blue. It's obviously dependent because the Eleview is distributed in 10 cc aliquot, so the more of them you use, the more costly it becomes. The good news, in this study, we used an average of 16 mLs, so that would be the equivalent of 2 vials of the viscous agent. And at that point, at least in our practice, that would be less expensive than our current self-mixed methylene blue agent.
Perfect. Thank you, Mike, and congratulations once again for this nice paper.
My pleasure. Thank you.
Thank you.